I'm the project leader of the Lab on a Chip biophotonic sensor platform for diagnostics. We are a team consisting of researchers and students from 3 different faculties at NTNU. We're from information technology and electrical engineering, natural sciences, medicine and health. And we also have people from SINTEF Digital. So we're a highly multidisciplinary team. So our project has become especially relevant in these pandemic times. So the need for fast diagnosis, I don't think anyone needs to be reminded of that just now. So not only increases the long waiting time, the risk for the patient's health, but we also get societal and economical implications that we're all aware of. So the project here is looking upon making a lab on a chip so that rather than having to send your blood samples or your urine samples to a central lab for analysis, that you can do it on a lab, on a chip, on a little chip. As I mentioned, we are a multidisciplinary team. We are people with different background. We have some people working with nanophotonics, some people working with functionalization of the sensor. We also work with nanofluidics and surface modification and conjugation. So what we're trying to do is we're trying to make a label-free sensor platform in order to perform both sensitive and selective multiplex quantitative diagnosis. What we want to be able to do is to analyze different biomarkers on the same chip, and to do this fast. So the goal of the project here is to develop a proof of principle demonstrator for research and potentially for treatment in a clinical environment. So what we do is that we use a silicon on insulator. Oops, sorry, wrong way. A silicon on insulator platform, and the reason we do this is that silicon on insulator is compatible with CMOS devices, so you have the potential to mass produce the chip part. The readout part currently in its form would be maybe a tabletop device, so that you could have disposable chips for each analysis. The first thing we had to do in this project is to develop the processing platform. So we've spent quite a lot of time and effort in being able to fabricate very small devices. They're on the order, what we're seeing here is micron. We are down in nanometer sized devices. And we can do this with high accuracy. So we have a really excellent, we use the Nano Lab at NTNU, and we have excellent equipment there, and especially our EBL, electron beam lithography equipment, is fantastic. So that was the first thing we had to do. And then we had to build up a setup to be able to characterize both the chips and also to perform measurements, because we're measuring concentrations of different biomarkers. So we identified 3 biomarkers that we wanted to investigate, and we were trying to press the limits of what we can achieve with regards to sensitivity. So this is just a setup we have in the lab, where we can both, where we put the chip, which is on this little table here, and then we couple light in and out, and we use microfluidics to stream the sample across the sensor. We started off looking at optical ring resonators to be which we functionalize. So what we do is we put these, this here is just a drawing of a simulation. So first we made the design by using FEM and FDTD, and then we functionalized the surface so that they're sensitive to these particular biomarkers. So the first thing we started measuring was CRP, C-reactive protein. And what happens is when the CRP binds to the antibody, then you get a shift in resonance. So here we see just, it's the principle of how the sensor works. It's sensitive to change in refractive index. So compared to if you have a fluid without the biomarker in it, you will not get a shift. But if your surface, since you have these antibodies which capture the biomarkers, then you get a change in refractive index. This is one of the first devices we made. So this is a ring resonator, and we started off measuring saltwater concentrations, and to see if we were able to achieve a linear response to with regards to a change in the concentration. So here's just a measurement where we're just showing that we get, we do indeed get, I have a plot also that shows a linear response. So it's a 6%, saline 12, etc., 18%. So this results in a shift in the resonance frequency of this ring resonator. Here I'm just showing the chemistry that we use. We silanize the surface of the ring resonator and then attach functional groups, and then you attach the antibodies, and these capture the proteins which we're measuring. Here's just another measurement where we see a shift in the resonance frequency when we flowed 5 micrograms per milliliter of CRP. Now it seems as though, there it goes. We've had many different types of sensor configurations. So we work with a typical ring resonator. We've also worked with photonic crystal ring resonators, and these are much smaller. So you could potentially have a larger number of sensors on the same little chip. So if we compare here, the ring resonator, which is 30 micron radius, which is a typical size you would have, then you have 900 microns squared of sensor area, whereas if you have a photonic crystal ring resonator, you can make these very small. But when you compare the sensitivity, you would have a higher sensitivity to the sensor, which has a larger sensor area compared to this photonic crystal. But still, you have quite a high sensitivity. Here, we're just showing measurements when we're flowing salt water and CRP. We're cleaning it with glycine, flowing salt water, and then measuring CRP. So this is just how we perform the measurements. We've developed a new configuration, which we call the MARC, which is a Mach-Zehnder interferometer assisted ring resonator configuration. We're currently we're patenting this configuration. It's quite special, and the reason we're using it is that we can make we can by changing the angle of the drop port here, we can make a unique spectral signature so that we can multiplex several or a number of resonators in the same channel and be able to differentiate the measurement of the different sensors using the same channel. So this can be quite useful when you're trying to make an inexpensive product. And these, so to the right here, we're just showing how by changing the angle between the through port and the drop port that you'll get a change in the phase with respect to when you're sweeping the wavelength across. So by doing this in a special way, you can generate these signatures. So here we just see the spectral response with different angular separations. And you see that the signatures are highly different. Here we've fabricated this MARC resonator. This one here has a 90 degrees phase shift, I believe. Yeah. We've also been developing the micro and nano fluidics for the sensor. So we have been making microfluidics using both PDMS, and recently we've developed processes for ostomer so that we can fabricate. Ostomer is also a polymer, and the advantage of using ostomer is that we can integrate passive mixers, which we put in the channel roof of the channel. And the reason we're using these passive mixers is to increase the mixing of the blood and the biomarkers, and try to sort of focus or push the biomarkers down onto the surface of the sensor. So it's to increase the sensitivity that we want these passive mixers. With PDMS, it's difficult to bond to the sensor and produce these structures, whereas we can likely do this in a good way with ostomer. So here we have some designs showing different passive mixers to increase the mass delivery to the biosensor surface. This is just a housing clamping system where we can put the little, which we use for test purposes, where we put our little chip in here. And this is a prototype of one of the ostomer microfluidic.
1 minute, Astrid.
Yeah, 1 minute. Okay, I'm almost finished. We're also, this is just measurements of where we have confocal images taken of different designs of passive mixing structures. So we have herringbone mixing structures and we have curved mixing structures. And to the right is a comparison where we see that CMS, that stands for the curved mixing structure, and HBS stands for herringbone. We see, what we can see here is that with short distances, you get better mixing with the curved mixing structures. We might be patenting these as well. So during the course of the project, we've spent a lot of time with process development, optimizing the microfluidics, optimizing the SOI platform. We've also with functionalization, and we've measured concentrations of CRP and cod VTG, a protein, in the nanogram per milliliter. And we have two rather novel designs, which have as a result of the project. So I think I will stop here. And I would like to thank all the partners in the project, all the students, and the Research Council of Norway for funding our project. And of course to Digital Life Norway Center for your support.